Hello and welcome back to Biology in Focus. Today we're going to work on Chapter 32, Homeostasis and Endocrine Signaling. My name is Mr. Sparks, and this is a lecture for John Tyler Community College. This lecture is going to get pretty in-depth towards the end when we start to talk about the functions of the kidney, so I just want you to be prepared for that and have your textbook available. There are diverse forms and common challenges to regulating the physiology of various organisms. Anatomy is the study of the biological form of an organism, and physiology is the study of the biological functions that an organism performs. Form and function are closely interrelated. The example we give here is one of the desert ant, which occurs in the Sahara Desert, perhaps one of the driest and most inhospitable regions on the planet. This ant is a scavenger, and it will go out and find the bodies of gerbils or snakes or other insects that have died on the desert sands. The desert sands in this region can get in excess of 140 degrees Fahrenheit. So it's just absolutely amazing temperatures there, and because of the lengths of the legs of the desert ant, it's able to keep its body up off the surface of the sand, and that may give it a reduced temperature of 6 degrees Celsius. So based on its function, its form, it's able to function in that intense environment. Okay, so all animals must obtain nutrients to obtain and oxygen and fight off infection and produce offspring. They all have these physiological functions. These desert ants can run as fast as 1 meter per second, so they're extremely rapid, and that reduces the time that they're exposed to the high temperatures on the surface of the soil. When they're not on the surface of the soil, they go back to their burrows, and it's much cooler inside those areas. But a lot of other organisms die in these temperatures, and the ants are able to capitalize on that by scavenging off of their corpses. In this chapter, we'll begin our study of animal form and function by examining the organization of cells and tissues in the animal body, the systems for coordinating the activities of different body parts, and the general means by which animals control their internal environments. In the second half of the chapter, we'll apply these ideas to 2 challenges of particular relevance for desert animals: regulating body temperature and maintaining proper balance of salts and water. So we're going to get into the homeostatic variables of these functions. Okay, in our first concept, we're going to look at the feedback control that maintains internal environment in many animals. Multicellularity allows for cellular specialization with particular cells devoted to specific activities. These are the various tissues. Specialization requires organization and results in internal environment that differs from the external environment. These are the functions of the organs and the organ systems. The hierarchical organization of animal bodies. Cells form functional animal body through emergent properties that arise from levels and structural functional organization. Cells are organized into tissues, which are groups of cells with a similar appearance and common function. Organs, which are different types of tissues organized into functional units. And organ systems, which are groups of organs that work together. Okay, here's the table 332.1 on page 642. It goes over the major organ systems, their main components, and main functions. I urge you to take a closer look at this table. Today we're going to go over the the endocrine system and the excretory system in greater detail. But these are all these different systems: digestive system, circulatory system, respiratory system, immune and lymphatic system, excretory system, endocrine system, reproductive system, nervous system, integumentary system, skeletal system, and muscular system. These are all different organ systems within vertebrates in general and mammals in particular. The specialized complex organ systems of animals are built from a limited set of cell and tissue types. Animal tissues can be grouped into 4 categories: epithelial, connective, muscle, and nervous tissues. Okay, here we have a vertebrate, a mammal, and these are the different tissues that compose the organs and the organ systems. Okay, I'm going to go into these. I'm going to go into these in detail here. The epithelial tissue is the tissue that occurs on the exterior surface of the organs. So epithelial tissue could be the skin or the dermis, which is the tissue which interfaces with the outside world. Okay, it may in this mammal, it's producing hairs that are helping to protect it from environmental variables. But then also on the inside of the animal, in the various different organs and organ systems, they have these tissues, the epithelial tissues that separate them from the other organ systems. And on the outside of these organ systems is where you have the epithelial tissue, protecting it from the other organs, separating it from the other organs inside the body. And then on the inside of those organ systems, like the digestive system in particular, you've got these epithelial systems, these epithelial tissues, which provide the surface area for the digestion of the various materials. When we go into the digestive tract, we're going to go into that in another chapter. That's where the epithelial tissue occurs. So on the inside and the outside of the digestive system, on the outside of the liver and the lungs, all right, there's epithelial tissue is found all over the place. Okay, then there's also the nervous tissue. The nervous tissue functions to receive and process exterior stimuli. You basically got the brain and the central nervous system are the 2 main parts of the nervous system. But nervous tissue occurs throughout the body. There are sensory nerves in every part of the body, particularly on the skin. And there's also nervous system tissue inside the body that helps to govern the autonomic nervous system, the nervous system that functions on its own, basically. The cardiac, the heart and lung system functions on their own, and the digestive system has nerves in it. But these are nerves that are separate from the nerves that act on external stimuli. These nerves act basically on internal stimuli and are self-regulating, but they all have similar function. The nerves here are shown in as yellow striations. These yellow tissues here are nervous tissue, and they are supported by glia. The glia are tissues that are wrapped up in around the nerves, the nerve functions, and they help support these different axons. Okay, these are the axons of the neurons here, and the glia are the supportive tissue that's surrounding that nervous tissue. Then we also have the connective tissue, which, interestingly, the loose connective tissue, which forms around the surface of that forms the structure of many organs. These have elastic fibers in them. They have collagenous fibers, which are fibers that collagen is a protein that helps to bind these tissues together. And then we also have the connective tissue, the blood. So blood is liquid connective tissue, and it carries plasma white blood cells, and which can consume they consume invasive tissues or invasive particles, bacteria or viruses, and they are part of the immune system. And red blood cells, which can transport oxygen and carry carbon dioxide to and from the tissues in the body. So those are two forms of connective tissue. Then the fourth form of connective tissue is the muscle tissue, and striated muscle tissue occurs in the skeletal muscles. There are looser structured muscle tissues that occur in the digestive system, which account for the smooth muscle activity that helps move food down from the mouth, from the pharynx into the digestive tract. Okay, and then it gets in here. It moves along through involuntary muscle structure in through the intestine, and then eventually exits the body as solid waste. Okay, so here's the epithelial tissue in a little more detail. This is the apical surface. This is where might be the inside of the lumen. This is the hollowed out area where the intestine opens up and forms a tube. This is the lumen area in here on this side, and then on the other end you've got the other side of the epithelial tissue. So the epithelial tissue shows greater structure, and it's affiliated with the interior of the intestine. Okay, axons of the nervous tissue. Here are the axons of the neurons. They're supported by the glia, which are basically helping to supply nutrients to the axons, and there are little blood vessels that run through this mechanism, providing blood for the axons of the neurons. This might be in the brain or central nervous system. Okay, here's loose connective tissue, like would be found on the surface of on the structure of the stomach. It's got elastic fibers and collagen proteins that allow it to move around, and so the loose connective fiber can also. This might include the loose connective fiber can include which holds skin and other organs in place. It's fibrous connective tissue found in the tendons and ligaments. It's adipose tissue, which stores fat, blood, and consists of cells and cell fragments suspended in a liquid called plasma. Cartilage, which supports flexible support in the spine and elsewhere, and bone, which is a hard mineral, calcium, magnesium, and phosphate ions in a matrix of collagen. So connective tissue takes a variety of different forms. Okay, here's the blood. The blood is the connective tissue. It's got the plasma, the white blood cell, the red blood cells. These are all different forms of blood, which are also considered connective tissue. Here's the skeletal muscle tissue. You can see the striated muscle cells in here. You can observe the various nuclei of that are forming the basis of these muscle cells. The skeletal muscle tissue, vertebrates have three types of muscle tissue: skeletal, smooth, and cardiac. All muscle cells consist of filaments containing the proteins actin and myosin, which together enable muscle cells to contract. Attached to bones by tendons, the skeletal muscle is striated muscle is responsible for voluntary movements. The arrangement of contractile units along the cells give the striated or appearance. Smooth muscle, which lacks striations, has spindle-shaped cells, which is found in the walls of many internal organs. Smooth muscles are responsible for involuntary activities such as the churning of stomach and constriction of arteries. Cardiac muscle is striated like skeletal muscle, and it forms the contractile wall of the heart. And the cardiac muscle is governed by the pacemaker. Faced with environmental fluctuations, animals manage their internal environment by either regulating or conforming. An animal that is a regulator uses internal mechanisms to control internal change despite external fluctuation. An animal that is a conformer allows its internal condition to change with accordance with external changes. Okay, so here's a river otter, which is a mammal. It regulates its body temperature by virtue of homeostatic regulation. It's got hair, and it produces its own body heat through metabolism, and so it's able to maintain its body temperature at a constant rate despite the variations in the ambient or environmental temperature. A bass, which is a fish, actually it has its temperature conforms to the ambient temperature. So if ambient temperature is increasing, then body temperature is also increasing. And just because an animal is able to maintain its own body temperature, or it's a conformer, or it's a regulator, that doesn't necessarily govern all of its homeostatic operations. So the bass is a temperature conformer, but it's able to regulate salts in its body based on its own internal metabolism. So an animal may regulate some internal conditions, but not others. For example, a fish may conform to surrounding temperature in the water, but it regulates solute concentrations in its blood and interstitial fluid, the fluid surrounding the body cells. Organisms use homeostasis to maintain a steady state or internal balance regardless of external environment. In humans, body temperature, blood pH, and glucose concentration are each maintained at a constant level. Regulation of room temperature by a thermostat is analogous to a homeostasis. So when humans regulate body temperature, blood pH, and glucose concentration, if you have the organ that regulates glucose concentration is the pancreas, and if you have problems with your pancreas, that might relate to diabetes. So impaired function of the glucose of the pancreas, it gives you the diabetic function, which means you're not unable to regulate glucose in your body. Okay, so here's a simple non-living example of temperature regulation and control of room temperature. Okay, here you've got your thermostat, which is set at 20 degrees Celsius. I don't know what that is. That's like 76 Fahrenheit. If the room temperature decreases, the thermostat kicks on the heater. The heater increases the room temperature, and then the room temperature is at 20 degrees Celsius. So the room temperature is at the desired level. This temperature increases. The sensor then turns the heater off. Okay, so turns the heater off, and the room temperature decreases. As the room temperature decreases, the room temperature's the set point is at 20 degrees. If it goes below 20 degrees, then then it's going to kick the heater back on. Okay, so this is a simple feedback loop. Animals achieve homeostasis by maintaining a variable at or near a particular value or set point. Fluctuations above and below the set point serve as stimulus, and these are detected as a sensor or trigger a response. The response returns the variable to the set point. Now, the way the animals do this is through the production of hormones and which cause a particular metabolic response. Homeostasis in animals relies largely on negative feedback, a control mechanism that reduces the stimulus. Homeostasis moderates but does not eliminate changes in the internal environment. Set points and normal ranges for homeostasis are usually stable, but certain regulated changes in the internal environment are essential. Okay, we take a look at thermoregulation. Thermoregulation is the process by which animals maintain an internal temperature within a tolerable range. This is the one that's easiest to conceive of because your temperature, you can obviously feel the changes in temperature differential, and it's easy to conceive of how thermoregulation works. Endothermic animals generate heat by metabolism, and birds and animals are endotherms. Ectothermic animals gain heat from external sources, and ectotherms include most invertebrates, fishes, amphibians, and non-avian reptiles. Endotherms can maintain a stable body temperature and the face of fluctuation in an environmental temperature. Ectotherms may regulate temperature by behavioral means. Ectotherms generally need to consume less food than endotherms because their heat source is largely environmental. So ectotherms, they can't really, they're not really as independent as endotherms. They can't really move great distances or whatever. They have to kind of stay in areas where the environmental conditions are good for them. This is why there's so many different species of reptiles in the tropics and the equatorial regions, because they need that heat source. And there are less diversity of reptiles in northern climates, but reptiles do occur in the northern climates, and those they rely, like rattlesnakes. They rely on just the summertime activities where they generate most of their energy by feeding on animals when they're able to come out above ground in the summertime, and then the wintertime they go below ground and hide out and conserve their resources, even though it gets to below freezing conditions. Like a study was done on rattlesnakes in Montana, and they were able to determine that a rattlesnake needs a minimum of 2 rats per year. It consumes 2 rats per year, and then it's able to survive. And then if it was able to eat up to 4 rats per year, it would have enough energy to reproduce. So ectotherms don't really they don't need a lot of food to survive, because their heat source is largely environmental. Okay, I want to take a look at this slide. I'm going to get a drink of water here. Okay, I'm back. A walrus is an endotherm. It's got a big fat layer of blubber. Even though it lives in the freezing cold, it's able to maintain its own internal body temperature. And a lizard is an example of an ectotherm. Here we see a lizard basking on the sunlight, receives warmth from the sun and solar radiation, and maybe from the rock if the rock gets nice and warmed up. And the lizards, a lot of reptiles like to stay in like a very small home range size. They find an area that's conducive to their biological system, and they like to stick around in that same location. Walruses travel all over the place. They migrate. They use their tusks to dig around in the muck to turn up clams and bivalves, which they feed on. So they have to eat constantly. Lizards eat insects as much as they can, but over the winter time they have to go find a nice little hole to hide in and try to survive the freezing temperatures of the winter time. They don't have a lot of activity in the winter time. Organisms exchange heat by 4 physical processes: radiation, evaporation, convection, and conduction. Heat is always transferred from the object of higher temperature to one of lower temperature. So radiation is like the energy that comes in from the sun. Evaporation is like when you sweat. That provides evaporation. That process of evaporation cools the surface of your skin. Convection is heat through the air, and conduction is direct from heat will be transferred from conduct from the organism to a surface. So an ectotherm can get heat from a warm rock, or you could lose heat by being in contact with the soil. Okay, so here's our reptile, our iguana. It looks like it could be a Galapagos marine iguana. It's getting heat through conduction, convection through the air. It's getting maybe cooling the iguana at this place. Evaporation, okay. Evaporation. Iguanas don't really sweat, but if it could get wet, it could get rained on, or could get swim in the ocean. Then that water would evaporate, and that evaporation would cool the iguana. Okay, and then there's radiation from the sun. So these are ways that an organism can get heat to help it thermoregulate. Circulatory adaptations for thermoregulation. In response to changes in environmental temperature, animals can alter blood and heat and flow between their body core and their skin. Vasodilation is the widening of the diameter of superficial blood vessels, and that promotes heat loss. And vasoconstriction is the narrowing of the diameter of superficial blood vessels that reduces heat loss. So in vasoconstriction, it's basically your capillaries and your smaller blood vessels are narrowing down, and basically that makes blood flow go to your internal organs. Vasodilation is the opposite of that, and so under cold conditions you have vasoconstriction. Under warm conditions you have vasodilation. The arrangement of blood vessels in many marine mammals and birds allows for countercurrent exchange. Countercurrent heat exchangers transfer heat between fluids in opposite directions and reduce heat loss. Okay, the example they give here is the Canada goose. I've seen the example with penguins. They do this too. The blood flow from the body of the penguin is a normal 35 degrees Celsius, so like 98, 100 degrees Celsius, or 98, 100 degrees Fahrenheit. So the warm blood comes from the body of the animal. It goes down to the feet. As it reaches the bottom of the feet into the toes of the organism here, in this case the Canada goose, it loses heat and then it cools down, but as it rises back up to the body, you don't put cold blood back into the body. That would shock the system. So that blood has to get warmed up somehow. So it gets warmed up through this countercurrent heat exchange. And by the time the blood it goes into a blood vessel, a vein, a returning vein. So it comes down through the artery, returns through the vein. The vein is next to the artery, and that allows it to warm up back into a normal temperature. Okay, birds and mammals can vary their insulation to acclimatize to seasonal temperature changes. Birds and mammals, birds will molt in the springtime. They'll lose all of their winter feathers, and they'll have they'll have less insulating feathers on their bodies during the winter time. The same can be said for mammals. Mammals will vary their insulation over the winter. During the winter time, the pineal gland, which is a gland deep in the brain, which we're going to talk about later on in this chapter, it senses the temperature. It senses the difference in the available sunlight, and after the winter solstice, it will induce the mammals to gain more fur, more underfur, and that insulation will acclimatize them to seasonal temperature changes. Acclimatization in ectotherms often includes adjustment at the cellular level. Some ectotherms that experience sub-zero temperatures can produce antifreeze compounds that prevent ice formation in their cells. There's a flounder that exists in the Antarctic regions. It exists in sub-zero conditions underneath the sea ice there, and it's able to produce antifreeze compounds that prevent ice formation in their cells. Interestingly, genetically modified tomato has been developed with the gene that is used by these flounders to produce their antifreeze, and it's helps the tomatoes to transport across great distances. So you can grow your tomatoes in Argentina and ship them up to New Jersey in the middle of the winter time. As for me, I just prefer locally grown tomatoes. I only eat tomatoes in the summertime when they're locally available. Thermoregulation in mammals is controlled by a region of the brain called the hypothalamus. The hypothalamus triggers heat loss or heat generating mechanisms. Fever is the result of a change to the set point for the biological thermostat. Okay, here we see some thermostatic function of the hypothalamus and human thermoregulation. We'll start here with the stimulus of the increased body temperature. That causes the thermostat in the hypothalamus to activate cooling systems. The response is that the sweat glands secrete sweat, which evaporate, cooling the body. Also, the blood vessels in the skin begin to dilate, and the capillaries fill with warm blood, which radiates heat from the skin surface. So this is the way that your body attempts to release heat into the environment. Body temperature decreases, the thermostat shuts off cooling mechanisms. Then we go back into the internal part of this mechanism, which is homeostasis. The internal body98.5 is 36 to 38 degrees, so 98.5 Celsius is 98.5 Fahrenheit is internal body temperature. Okay, so then we go to the stimulus where it decreases body temperature. The decreased body temperature, such as when in cold surroundings. The sensor control center, the thermostat, and the hypothalamus activates warming mechanisms. This can cause the blood vessels to constrict, the capillaries to constrict, and the Blood vessels in the skin constrict, diverting blood from the skin to the deeper tissues and reducing heat loss from skin surface. The response could be shivering, which is skeletal muscles rapidly contracting and causing shivering, which generates heat, and body temperature increases in the thermostat, shuts off the warning mechanisms. Okay, so basically, if you go below 98.5, you're going to cause this pathway to occur. Constriction of the capillaries and the blood vessels, and the shivering response, and that's going to cause the body temperature to increase. And if you obtain your increase back to 98.6 Fahrenheit, then you're going to be maintaining homeostasis. If you don't maintain homeostasis, then you go back into the cycle where it's going to try to increase your body temperature again. It's going to do this until it attains body temperature, normal body temperature, and then the opposite stimulus occurs with increased body temperature. Okay, shivering and chills, like if you get malaria, you get chills and then you get shivering. That's basically your body is responding to these different stimulus, internal stimulus that are caused in the hypothalamus. It's basically your body deceiving itself. Okay, so here we go. Increased body temperature, sweat glands, dilating the blood vessels, increases body temperature, decreases, back down to normal. Okay, body temperature decreases. All right, vasoconstriction, shivering, body temperature increases, back to homeostasis. The endocrine signals trigger homeostatic mechanisms in target tissues. There are two major systems for controlling and coordinating responses to stimuli: the endocrine and the nervous system. The endocrine system signaling molecules released into the bloodstream by the endocrine cells reaches all locations of the body. Basically, the master gland is located in the center of the brain, called the hypothalamus, and we're going to go into that in greater detail in the next part of the chapter. In the nervous system, neurons transmit signals along dedicated routes connecting specific locations in the body. Signaling molecules sent out by the endocrine system are called hormones. Hormones may have effects in a single location throughout the body. Only cells with receptors for a certain hormone can respond to it. So hormones are typically released into the bloodstream and they travel all throughout the body, but only glands with the receptors for the certain hormone can respond to it. The endocrine system is well adapted for coordinating gradual changes that affect the entire body. Okay, here are signaling in the endocrine and the nervous system. Signaling by an endocrine cell may release hormones into the blood system, and then through the capillaries, these hormones are picked up only by cells that have the specific receptor cells, and then they're going to give the response. Okay, they cause that gland or that organ to release release other hormones or release other compounds that are necessary for homeostasis. The other route is through neurons. So stimulus can reach the neurons. They can travel through a specific location, and they may cause this would be like an example would be the release of adrenaline into the. You may have a traumatic experience. You may get cut with a knife, and you're in a knife fight, and you've got to release adrenaline in order to survive. That would cause the nerve impulse to release to the adrenal glands, and that would release adrenaline into the blood system, which would cause a number of responses in your body to enable you to respond to that situation. Okay, this is on page 648. In the nervous system, signals called nerve impulses travel along communication lines consisting mainly of axons. We are going to go into the nervous system in chapter 28, which is the last chapter of this unit. So that's going to be, we're going to do that in a little more detail. Other neurons, muscle cells, and endocrine and exocrine cells can all receive nerve impulses. Nervous system communication usually involves more than one type of signal. The nervous system is well suited for directing immediate and rapid responses to the environment. Okay. So those, like I was talking about, like adrenaline, like signaling by hormones is more like maintaining homeostasis in your glucose levels through your pancreas, or as compared to like an immediate fight or flight response through the adrenal glands that was caused by a nerve impulse. Simple endocrine pathways. Digestive juices in the stomach are extremely acidic and must be neutralized before the remaining steps of digestion take place. Coordination of the pH control in the duodenum, or some people say duodenum, relies on the endocrine pathway. Okay, so basically what happens is you're in your duodenum, which is the first part of your lower intestine, which occurs just outside of the stomach. Okay, where the stomach empties into the small intestine. That's the region called the duodenum. Your duodenum is sensitive to low pH, so it needs to reach a more normalized pH, pH closer to 7. Usually the pH inside your stomach is like a pH of 2 to 4. So it's a pretty low acid, hydrochloric acid content there. It needs to be reduced once it reaches into the duodenum. So when the stomach churns out and releases the chyme, the stomach content into the lower intestine, into the duodenum, it kicks off the stimulus, which in the endocrine cell in the duodenum. So these S cells, they will secrete the hormone secretin. This hormone secretin then goes into the blood vessels, and it reaches the target cells, which are in the duodenum or in the pancreas, and the pancreas releases bicarbonate. And bicarbonate modifies the pH level of the chyme. So then that is the response, and then it reduces the pH of the chyme. Now, if that pH is still low, then it will cause that stimulus to go over and over again until enough bicarbonate is released to where the duodenum senses that it's and the proper pH level. Okay, until that pH level is reached, that response and stimulus is going to go over and over again, releasing bicarbonate from the pancreas. The release of acidic stomach contents into the duodenum stimulates endocrine cells there to secrete the hormone secretin. This causes the target cells of the pancreas to raise the pH of the duodenum through the release of bicarbonate. The pancreas can act as an exocrine gland, secreting substances through a duct, or as an endocrine gland, secreting hormones directly into the interstitial fluid. Neuroendocrine pathways. Hormone pathways that respond to stimuli from the external environment rely on a sensor in the nervous system. In vertebrates, the hypothalamus integrates endocrine and nervous systems. Signals from the hypothalamus travel to a gland located at its base called the pituitary gland. The pituitary gland is the hypothalamus is the master gland, and the pituitary gland is the gland that releases all these different kinds of hormones that will stimulate different activities in the human body or in the vertebrate body. Okay, here are the major endocrine glands and their hormones. The endocrine system, it produces hormones that regulate growth, development, metabolism, homeostasis, and reproduction. Many hormones are secreted by the ductless organs called the endocrine glands, illustrated below. Others are secreted by isolated endocrine glands in other organs, the thymus, the heart, liver, stomach, small intestine, and kidneys. Regardless of where the hormones are produced, they reach their target cells via circulatory system. Okay, so the hypothalamus and the pituitary gland are considered the master gland. Now the pineal gland, which I mentioned earlier here, the pineal gland is located right in the center of the brain. This is the organ that it actually senses variations in light over the seasons. So in fur-bearing mammals, when the winter solstice occurs, the pineal gland releases compounds that hormones that tell the body of the mammal, of the fur-bearing mammal, to produce more fur, produce a higher quality fur to prepare for winter conditions. It's also implicated with melatonin, which is a hormone that's involved in the circadian rhythm. You know, people that travel a lot, they get jet lag. They travel from one coast to the other, or they travel to Europe a lot. They get jet lag through. It basically confuses the circadian rhythm. Circadian rhythm is your daily biological function. So you get jet lag by traveling too much. Some people take a supplemental of melatonin that supposedly helps them to adjust with those capacity. But here's the pineal gland. Its function is to help moderate the circadian rhythm. Now here's the hypothalamus and the pituitary gland. These are the master gland. These glands, they produce all kinds of different hormones. They produce follicle-stimulating hormone, thyroid-stimulating hormone, adrenocorticotropic hormone, prolactin, melanocyte, growth hormone. Okay, they produce all different kinds of hormones that affect these other endocrine glands. So they may release the growth hormone, which has a impact over all the different parts of the human body. So growth hormone is particularly active up until you're about age 17 or 18. Pituitary gland is producing growth hormone until you reach your adult phase. Okay, the pituitary gland is located right there, just behind the eye. The posterior pituitary produces oxytocin. Oxytocin is also known as the cuddling hormone. It's the hormone that gives you a sense of emotional security. Vasopressin, the antidiuretic hormone, which we're going to get into when we talk about the kidneys in the next couple of chapters here. There are the adrenal glands, which sit on top of the kidneys. These are the glands that are responsible for the release of adrenaline, which can be stimulated during emergency situations. Then there's the the pancreas, which produces insulin and glucagon, which are hormones responsible for moderating the level of sugars in our bodies. And then there are the ovaries in the females and the testes in the males. These are producing hormones related to estrogens and progesterones. They are hormones related to the production of eggs in the reproductive system. We're going to talk about that later on in this unit in another chapter. Then there is the parathyroid and the thyroid hormones. These release compounds that are related to condition of the thyroid. Okay, so they all have these different functions, but most of them are affected by the pituitary gland and the hypothalamus. Okay, here's the hypothalamus, the roles of the hypothalamus and the anterior pituitary. Hormones produced by the hypothalamus regulate the anterior pituitary. The hypothalamic hormones are secreted near the capillaries of the base of the hypothalamus. These vessels, which connect directly to the secondary capillary bed in the anterior pituitary, hypothalamic hormones thus travel from hypothalamus to the gland. They regulate with first circulating through the other body tissues. Upon reaching the anterior pituitary, each hypothalamic hormone either stimulates or inhibits the release of one or more specific hormones. So these are all the follicle-stimulating hormones which affect the testes and the ovaries, thyroid-stimulating hormones which affect the thyroid, the adrenal cortex, adrenal corticotropic hormones which affect the adrenal cortex, prolactin which affect the mammary glands, melanocyte-stimulating hormones, the liver, bones, and other tissues, growth hormones. All these different hormones are stimulated. That the anterior pituitary is the master gland, so it produces the hypothalamus, and the master pituitary and the anterior pituitary are the master glands. They work together through different releasing of different neurotransmitters and hormones within them. They generate different hormones, and that affect all of these different endocrine glands. Okay, here are the different hormones that are produced by the hypothalamus and the anterior pituitary. Okay, here's a hormone cascade pathway. The sets of hormones in the hypothalamus, the anterior pituitary, and the target endocrine gland are often organized into a hormone cascade pathway. Signals to the brain stimulate the hypothalamus to secrete a hormone that regulates the release of an anterior pituitary hormone. The anterior pituitary hormone, in turn, acts on another endocrine gland, stimulating the secretion of yet another hormone, which exerts effects on specific target tissues. Because each pathway, in a sense, redirects signals from the hypothalamus to another endocrine pathways, the anterior pituitary hormones of these pathways are called tropic hormones or tropins, from the Greek word for bending or turning. One example of a hormone cascade pathway is the regulation of the thyroid hormone levels shown here. Okay, so here the thyroid hormone drops below the normal range. Okay, we have our target range is our homeostatic state. but in this case, the thyroid hormone is dropping below that value. Thyroid hormone levels drop below the normal range. The hypothalamus secretes thyrotropin releasing hormone, which into the blood, which through the portal vessels which carry the thyrotropin releasing hormone into the anterior pituitary. Okay. Releases the hormone into the anterior pituitary. The TRH, the thyrotropin releasing hormone, causes the anterior pituitary to secrete thyroid stimulating hormone, also known as thyrotropin, into the circulatory system. Then it goes the thyrotropin, the thyrotropin stimulating hormone. The releasing hormone causes the stimulating hormone. The stimulating hormone is passed out into the bloodstream. It goes throughout the body, and it affects the thyroid gland, where the thyroid gland has the capacity to receive those hormone signals. The hormone signal then stimulates the endocrine cells in the thyroid to secrete the thyroid hormone T3 and T4 into the circulatory system. The thyroid hormone levels then increase in the blood through the body tissues, returning to the normal range. Thyroid hormone acts on target cells throughout the body to control bioenergetics. To help maintain normal blood pressure, heart rate, and muscle tone, and regulate digestive and reproductive function. So the thyroid hormone helps to regulate the circulatory system, and it gets back into the bloodstream where it goes. It's either if it's a reduced amount of it, you're not having normal circulatory function. The hypothalamus stimulates the anterior pituitary, releases the hormone into the bloodstream. It affects the thyroid, which releases the endocrine hormones necessary to maintain homeostasis in the circulatory system. Then it goes back through the system where if it's low, it causes more stimulus. If it's normal, then the stimulus is satiated. Okay, here's a thyroid gland. There's one has low iodine uptake, other has high iodine uptake. These are radioisotopes of iodine, and it shows how you can discern the dramatic different activity of the two lobes of the thyroid gland in a patient with thyroid disease. Okay, hormonal signals from the hypothalamus trigger synthesis and release of hormones from the anterior pituitary. The posterior pituitary is an extension of the hypothalamus, which secretes oxytocin, which regulates the release of milk during nursing in mammals. It also secretes antidiuretic hormone. Okay, so here's an example. Any of you who are mothers probably are familiar with this. A baby suckling can cause the stimulus. This is a particular positive feedback loop, which is different than the other feedback loops, and it causes stimulation of the hypothalamus, which releases the neuroendocrine cell, releases the neurohormone oxytocin. Oxytocin stimulates the blood vessels in the breast to relax the smooth muscles in the breast and release the milk. This is commonly known as milk letdown. So this is the path that causes the milk release. And then that suckling stimulation will continue to cause the oxytocin to be released, and then that oxytocin will that oxytocin will then cause more milk to be released, and basically this stimulus will go in a positive feedback loop until there's no milk remaining in the mammary glands. And then at that point, it's the end of the milk. But up until that point, it's just a positive feedback loop. The more suckling, the more releasing of the milk until it's been reduced. Okay, feedback regulation and endocrine pathways. A feedback loop links the response to the original stimulus in the endocrine pathway. When negative feedback dampens a stimulus, positive feedback reinforces a stimulus to increase the response. Pathways of water-soluble and lipid-soluble hormones. The hormones discussed thus far are proteins that bind to cell surface receptors and trigger events leading to cellular response. Intracellular response is called a signal transduction. A signal transduction pathway typically has multiple steps. Lipid-soluble, which are fat-soluble hormones, have receptors inside cells. When bound by the hormone receptor complex moves into the nucleus. There, the receptor alters transcription of a particular genes. Many hormones elicit more than one type of response. For example, epinephrine is secreted by the adrenal glands. It can raise blood glucose levels, increase blood flow to the muscles, and decrease blood flow to the digestive system. Target cells vary in their response to a hormone because they differ in their receptor types or in their molecules that produce the response. Okay, so here's epinephrine. It can cause blood vessels to dilate, the skeletal blood vessels, or it can cause, the same hormone can cause blood vessels to constrict. So basically you're in an emergency situation, you need more oxygen and glucose to your blood vessels. Your blood vessel, epinephrine causes them to dilate, but you don't need your digestive system. You need to shut down your digestive system in order to make more blood available to your skeletal muscle system. And then the same hormone, epinephrine, causes those blood vessels to contract. So the same receptors, but different intracellular proteins, okay, can cause different responses. The evolution of hormone function. Over the course of evolution, the function of a given hormone may diverge between species. For example, thyroid hormone plays a role in metabolism across many lineages, but in frogs it has taken on a unique function, stimulating the reabsorption of the tadpole tail during metamorphosis. Prolactin also has a broad range of activities in vertebrates. Okay, so here is the hormone thyroxine, which is responsible for the reabsorption of the tadpole tail into the frog as the frog develops into the adult form. There's a tadpole, there's a frog. A shared system mediates osmoregulation and excretion in many animals. Okay, now we're getting closer to the function of the kidney. Osmoregulation is a general term for the processes by which animals control solute concentrations in the interstitial fluid and the balance of water gain and loss in the cells. Cells contain a balance between uptake and loss of water. Osmolarity is the solute concentration of a solution, determines the movement of water across a selectively permeable membrane. If two solutions are isoosmotic, the movement of the water is equal in both directions. So where the amount of solute is the same in two different solutions, the direction of water travel is equal in both directions. That's called isoosmosis. If two solutions differ in osmolarity, the net flow of water is from the hypoosmotic to the hyperosmotic solution. So hypoosmotic, like a hypodermic needle, is below the skin. So hypoosmotic means it has less solute concentration. So the water will move from the hypoosmotic solution to the hyperosmotic solution. So it will move from the low solute content to the high solute content, because it's trying to get water in there to even out to reach an equilibrium. Osmoconformers, consisting of some marine animals, are isoosmotic with their surroundings and do not regulate their osmolarity. Osmoregulators, which is the vast majority of other animals, expend energy to control water uptake and loss in a hyperosmotic or hypoosmotic environment. Marine and freshwater organisms have opposite challenges. Marine fish drink large amounts of seawater to balance water loss and excrete salt through their gills and kidneys. Freshwater fish drink almost no water and replenish their salts through eating. Some also replenish salts by uptake across the gills. So the freshwater fish, it needs to retain salts in its body, and the marine fish needs to get rid of salts out of its body. Osmoregulation in a marine fish. Okay, so basically what we see here is the gain of water from salt and ions from food. Okay, the marine fish is trying to get rid of salts. It excretes salts ions from water and through the scanty urine in the kidneys. Okay, it's trying to retain fresh water in its body. And the freshwater fish, it gains some of the water from ions from food, uptake by salt ions in the gill, osmotic regulation, and excretes salt ions in large amounts of water through dilute urine in the kidneys. So it has dilute urine. It's trying to maintain salts inside, and this has a lot of urine. Okay, and this trying to maintain water inside the fresh water inside the system. Okay, so for the cod, the ocean is a strongly dehydrating environment. Constantly losing water by osmosis, such fishes balance the water loss by drinking large amounts of seawater. In ridding themselves of salt, they make use of both their gills and their kidneys. In the gills, especially, chloride cells actively transport chloride ions out and allow sodium ions to follow passively. In the kidneys, excessive calcium, magnesium, and sulfate ions are excreted in the loss of only small amounts of water. So the saltwater fish tries to retain fresh water. The freshwater perch lives in an environment with a low osmolarity, so it faces the problem of gaining water by osmosis and losing salts by diffusion. Like many freshwater animals, the perch solves this problem by drinking almost no water and excreting large amounts of fresh dilute urine. At the same time, salts by diffusion and in the urine are replenishing by eating. Freshwater fishes such as the perch also replenish salts by uptake across the gill. Land animals have mechanisms to prevent dehydration. Most have body coverings that help reduce water loss, except for the exception of the amphibians. They drink water and eat moist foods, and they produce water metabolically. Okay, nitrogenous waste. This is one of the most important wastes that are excreted. The type and quantity of an animal's waste products may greatly affect its water balance. Among the most significant wastes are nitrogenous breakdowns of proteins and nucleic acids. Some animals convert toxic ammonia into less toxic compounds prior to excretion. Okay, so fish excrete ammonia directly into the environment. They don't really have a high concern with ammonia, but in mammals and birds, they have to travel around and conserve their waste until the time when it's when it's a good time to release those wastes. So in the bodies of mammals, ammonia is converted into urea, and in birds it's converted into uric acid. Now, uric acid is like a largely dry waste. So, uric acid is the white part of a bird's poop, and the black part of the bird's poop is generally like the body of the insect or the animal that it's consumed. So that uric acid is their way of conserving water, and mammals need water to evacuate urea. But we can metabolize urea for a certain amount of time, so we don't have to go to the bathroom all the time. Ammonia excretion is common in aquatic organisms. Vertebrates excrete urea, a conversion product of ammonia, which is much less toxic. Insects, land snails, and many reptiles, including birds, excrete uric acid as a semi-solid paste. It is less toxic than ammonia and generates very little water loss, but it is energetically more expensive to produce than urea. So you just have urea, you can release it right away, but you don't. In order to make urea less toxic, it takes some metabolic activity. In most animals, osmoregulation and metabolic waste disposal rely on transport epithelia. These are the layers of epithelial cells which are specialized for moving solutes in controlled amounts in specific directions. Many animal species produce urine by refining filtrate derived from bodily fluids. Key functions of the most excretory systems are filtration, the filtering of bodily fluids, reabsorption, the reclaiming of valuable solutes, secretion, adding non-essential solutes and waste from the body fluids to the filtrate, and excretion, releasing processed filtrate containing nitrogenous waste from the body. Okay, here's a very simple schematic of what goes on in the kidney. First, there's filtration up at the top here. The excretory tubule collects a filtrate from the blood. Water and solutes are forced by blood pressure across the selectively permeable membranes and a cluster of capillaries into the excretory tubule. Okay. The reabsorption is the transport of the epithelium, reclaims valuable substances from the filtrate and returns them to the body fluids. Secretion is when other substances such as toxins and excess ions are extracted from the body fluids and added to the contents of the excretory tubule. Excretion is the altered filtrate, the urine, which leaves the system and the body. Most excretory systems produce a filtrate by pressure filtering bodily fluids and then modifying filtrate contents. This diagram is modeled after the vertebrate excretory system. So this is a very simple look at the kidney. We're going to get into the kidney in greater detail later on in this chapter. Invertebrates, flatworms have excretory systems called protonephridia, networks of dead-end tubules connected to external openings. They operate off the gastrovascular cavity. The smallest network branches of this network are capped by cellular units called a flame bulb. These tubules excrete a dilute fluid and function in osmoregulation. Okay, so here the protonephridia are branching internal tubules that function mainly in osmoregulation. So the wastes are going into in through the, here's the gastrovascular system in the center, and then the tubules of the protonephridia are surrounding that gastrovascular cavity, and then they're communicating. The waste compounds are going in through the flame bulbs. They have cilia inside here, which help to direct the waste into the protonephridia, and they go into the opening and out through the body wall. In insects and other terrestrial arthropods, Malpighian tubules remove nitrogenous waste from the hemolymph without a filtration step. Insects produce a relatively dry waste matter that is called uric acid. And if you go under a tree that has a lot of caterpillars in it, you'll notice that it has little brown dots, and these little brown dots are called frass, which is the waste. It's the mainly uric acid, dry waste material of the caterpillars. In vertebrates and some other chordates, the kidney functions in both osmoregulation and excretion. The kidney consists of tubules arranged in an organized array, which are in contact with a network of capillaries. The excretory system includes ducts and other structures that carry urine from tubules out of the body. In humans, the excretory system consists of a pair of kidneys, these bean-shaped organs about 10 centimeters in length, as well as organs from transporting as well as organs for transporting and storing urine. Urine produced by each kidney exits through the duct called the ureter, and the two ureters drain into a common sac called the urinary bladder. During urination, urine is expelled from the bladder through a tube called the urethra, which empties the outside near the vagina in females and through the penis in the males. Sphincter muscles near the junction of the urethra and the bladder regulate urination. Okay, I think I'm going to go into these slides in greater detail here. If not, we'll backtrack. But this is the kidney structure. There's basically the outside of the kidney is called the cortex. The inside is called the medulla. There's the renal artery and the renal vein, and then there's the ureter, which it collects urine and transports it to the bladder. Inside the kidney, on a closer look here, the cortical nephron. This is where waste is transferred from the renal artery into the glomerulus. Oh boy, this is really looks complicated, so I'm going to get into it in greater detail as we go along here. Okay, so this is the greater detail inside the kidney. You've got the glomerulus, the Bowman's capsule, which surrounds the artery, the renal artery, and this is where wastes are expressed into here through osmolytic properties into the urine here that's collected into the proximal stream. This is called the loop of Henle here, and it travels through here. Wastes are exchanged through the vasa recta, which goes back into the renal vein, and then it collects into the back into the proximal back into the tubule of the distal tubule, and then into the distal tubule. It goes into the collecting duct, into the ureter, and then and from the ureter into the bladder. I'm going to go into this in greater detail. Okay, so here is the kidney. Each kidney has an outer renal cortex and an inner renal medulla. Both regions are supplied with blood and by a renal artery and drained by a renal vein. Within the cortex and the medulla lie tightly packed excretory tubules and associated blood vessels. The inner renal pelvis collects urine from the excretory tubules and passes it through the urinary bladder. These are the nephron types. Weaving back and forth across the renal cortex and the medulla are the nephrons, the functional units of the vertebrate kidney. Most of the cortical nephrons, which reach only a short distance into the medulla. The remainder are the juxtamedullary nephrons, which extend deep into the medulla. Juxtamedullary nephrons are essential for the production of urine that is hyperosmotic to body fluids. The nephron organization. Each nephron consists of a single long tubule and a ball of capillaries called the glomerulus. The blind end of the tubule forms a cup-shaped swelling called the Bowman's capsule, which surrounds the I got ahead of myself there. So basically, here's your afferent arteriole from the renal artery. It goes in. It forms a capillary network in here. That capillary network is covered by the Bowman's capsule. The Bowman's capsule surrounds the glomerulus. The filtrate is formed when blood pressure forces fluid from the blood in the glomerulus into the lumen of the Bowman's capsule. That's the structure of the Bowman's capsule. Processing occurs as filtrate passes through the three major regions of the nephron: the proximal tubule, the loop of Henle, which is a hairpin turn in the descending limb and the ascending limb, and the distal tubule, and a collecting duct receives processed filtrate from many nephrons and transports it to the renal pelvis. Each nephron is supplied with a blood by an afferent arteriole, an offshoot of the renal artery that branches and forms capillaries of the glomerulus. The capillaries converge as they leave the glomerulus, forming an efferent arteriole. Branches of this vessel form the peritubular capillaries, which surround the proximal and distal tubes. Each other branches extend downward and form the vasa recta, hairpin-shaped capillaries that serve the renal medulla and surround the loop of Henle. So this is where your exchange of your waste material is occurring in the Henle. Basically, the loop of Henle is a very important part of that structure. It's surrounded by the vasa recta, which is exchanging through osmolarity. It's exchanging waste into the urine stream. Hormonal circuits link kidney function, water balance, and blood pressure. The capillaries and specialized cells of the Bowman's capsule are permeable to water and small solutes, but not blood cells or large molecules. The filtrate produced there contains salt, glucose, amino acids, vitamins, nitrogenous waste, and other small molecules. The proximal tubule. The reabsorption of ions and water nutrients takes place in the proximal tubule. Molecules are transported actively and passively from filtrate into the interstitial fluid and then to the capillaries. Some toxic materials are actively secreted into the filtrate. The descending limb of the loop of Henle. Reabsorption of water occurs through channels formed by aquaporin proteins. Movement is driven by the high osmolarity of the interstitial fluid, which is hyperosmotic to the filtrate. And the filtrate becomes increasingly concentrated all along its journey down the descending limb. The ascending limb of the loop of Henle. The ascending limb has a transport epithelium that lacks water channels. Here, salt but not water is able to move from the tubule from the interstitial fluid. The filtrate becomes increasingly dilute as it moves up to the cortex. The distal tube. The distal tube regulates potassium and sodium chloride concentrations of body fluids. The controlled movement of ions contributes to pH regulation. The collecting duct. Okay, the collecting duct carries the filtrate through the medulla into the renal pelvis. Most of the water and nearly all of the sugars and amino acids and other vitamins and other nutrients are reabsorbed into the blood. Urine is hyperosmotic to bodily fluids. Okay, so here's a schematic of the nephron and the collecting duct. These are the regional functions of the transport epithelium. The numbered regions in this diagram are key to the circled numbers in the text of the discussion of the kidney function. So you may want to follow along in your textbook on page 658. We're going to go through this whole thing. From blood filtrate to urine, a closer look. In this section, we will follow a filtrate along its path in the nephron and collecting duct, examining how each region contributes to the stepwise processing of filtrate into urine. Each circled number refers to the processing in a particular region, as illustrated in figure 32.2. The proximal tubule. Reabsorption in the proximal tubule is critical for the capture of ions and water and valuable nutrients. This is where salt is filtered into it enters into the cells of the transport epithelium by filtrated diffusion and co-transport mechanisms. Their sodium is actively transported into the interstitial fluid. This transfers the positive charge out of the tubule and drives the passive transport of chlorine. As salt moves from filtrate into the interstitial fluid, water follows by osmosis. The salt and water diffuse from the interstitial fluid into the peritubual capillaries. Glucose, amino acids, potassium ions, and other essential substrates are actively or passively transported through the filtrate into the interstitial fluids and then into the peritubual capillaries. In contrast, some toxic materials such as drugs and toxins that have been processed by the liver, are actively secreted into the filtrate by the transport of the epithelium. It's interesting to note that environmental scientists have been able to detect pharmaceuticals in our water supply because of the amount of pharmaceuticals that we excrete in our urine. And they can actually identify them by brand name. The descending loop of Henle. Reabsorption of the water continues as the filtrate moves into the descending limb of the loop of Henle. Here, numerous water channels are found by aquaporin, proteins that transport epithelium freely permeable to water. In contrast, there's almost no channels for salt and other small solutes, resulting in very low permeability for these substances. For water to move out of the tubule by osmosis, the interstitial fluids bathing in the tubule must be hyperosmotic to the filtrate. The condition here is met by the entire length of the descending limb because the osmolarity of the interstitial fluid increases progressively from the outer cortex to the inner medulla of the kidney. Consequently, the filtrate loses water and increases in a solute concentration along the journey of the descending limb. Now, in the ascending limb of the loop of Henle, the filtrate reaches the tip of the loop and then returns the cortex with the ascending limb. Unlike the descending limb, the ascending limb has a transport epithelium that lacks water channels. As a result, this region of the epithelium membrane faces the filtrate is impermeable to water. The ascending limb has two specialized regions: the thin segment near the loop tip and the thick segment adjacent to the distal tubule. The filtrate ascends the thin segment with sodium chloride, which became concentrated in the descending limb, diffuses out and the permeable tubule into the interstitial fluid. This movement of sodium chloride out of the tubule helps maintain the osmolarity of the interstitial fluid in the medulla. The thick segment of the ascending limb. The movement of sodium chloride out of the filtrate continues. Here, however, the epithelium actively transports sodium chloride into the interstitial fluid. As a result of losing salt but not water, the filtrate becomes progressively more dilute as it travels up the cortex and in the ascending limb of the loop. The distal tubule. The distal tubule plays the role in regulating potassium and sodium chloride concentrations in the body fluids. This regulation involves variation in the amount of potassium secreted into the filtrate, as well as the sodium chloride reabsorbed from the filtrate. Like the proximal tubule, the distal tubule contributes to pH regulation by the controlled secretion of hydrogen ions and the reabsorption of carbonic acid. The collecting duct. Collecting duct carries the filtrate through the medulla into the renal pelvis. Final processing of the filtrate by the transport epithelium into the collecting duct forms urine. Okay. Well, under normal conditions, about 1,600 liters of blood flows through the pair of human kidneys each day, processing the enormous amount of traffic of nephrons and collecting ducts, yielding about 180 liters of initial filtrate. Of this, about 99% of the water and nearly all of the sugars, amino acids, vitamins, and other organic nutrients are reabsorbed by the blood, leaving only about 1.5 liters of urine to be transported to the bladder. So an immense amount of fluid goes through the bladder, through the kidneys each day, but so much of the nutrients are reabsorbed through the bladder that and nutrients and water are reabsorbed by the bladder that only 1.5 liters of leaves the body as urine each day. Okay. All of this is on page 659, 658, 659 in your textbook. The mammalian kidney's ability to conserve water is a key terrestrial adaptation. Okay, so mammals are able to. This is how the human kidney concentrates urine, the two-solute model. Two solutes contribute to the osmolarity of the interstitial fluid. Sodium chloride, used here as in a shorthand to refer to sodium and chlorine in urea. And the loop of Henle maintains the interstitial gradient of sodium chloride, which increases continuously in concentration from the cortex to the inner medulla. The urea diffuses into the interstitial fluid into the medulla from the collecting duct. Most of the urea in the filtrate remains in the collecting duct and is excreted. The filtrate makes 3 trips down between the cortex and the medulla. First down, then up, and then down again in the collecting duct. As the filtrate flows in the collecting duct past the interstitial fluid of the increasing osmolarity, the more water moves out of the duct by osmosis. The loss of water concentrates the solutes, including urea, that will be excreted in the water. So this is how the water is transported out of the there's a high osmolarity in the inner medulla, and there's low osmolarity in the outer medulla. So the high osmolarity basically helps it causes the water to be passed out through the proximal distal, or through the loop of Henle here, and it goes from the urine, from the waste stream out into the medulla. So that's how water is extracted from the urine, and then the waste materials go down ultimately into the ureter. Okay, so as it goes up, water goes out, and as it goes back down, then solutes are expelled. So the useful sodium chloride, which is standing in for the waste here, or the nutrients, goes back into the. Okay, so there's aquaporons here that allow the water to be transport. There's no aquaporons here, which forces the sodium chloride to be extracted, and then on through the end here. That's where the okay. This is on page 660. Such a system which expends energy to create a concentration gradient is called a countercurrent multiplier system. Water and salt are reabsorbed from the filtrate, passing from the Bowman's capsule to the proximal tubule. In the proximal tubule, filtrate volume decreases, but osmolarity remains the same. As the filtrate flows down the descending limb of the loop of Henle, water leaves the tubule, increasing the osmolarity of the filtrate, and salt diffusing from the ascending limb maintains a high osmolarity in the interstitial fluid of the renal medulla. The loop of Henle and the surrounding capillaries act as a type of countercurrent system. This system involves active transport and thus has an expenditure of energy. Such a system is called the countercurrent multiplier system. The filtrate in the ascending limb of the loop of Henle is hypoosmotic to the body fluids by the time it reaches the distal tube. The filtrate descends the collecting duct, which is permeable to the water but not the salt. Osmosis extracts water from the filtrate and concentrates salts, urea, and other solutes in the filtrate. The adaptations of the vertebrate kidney to diverse environments. Variations in the nephron structure and function equip the kidneys of different vertebrates for osmoregulation in their various habitats. Desert-dwelling animals excrete the most hyperosmotic urine and have long loops of Henle. Birds have shorter loops of Henle, but conserve water by excreting uric acid instead of urea. So they have to metabolize that. Their waste is less metabolized. Mammals control the volume and osmolarity of urine. The kidneys of South American vampire bat can produce either very dilute urine or very concentrated urine, depending on whether or not they've just fed. This allows the bats to reduce their body weight rapidly or digest large amounts of proteins while conserving water. So they make. So here's the bat. If it was gonna fly immediately after it has its blood, the blood intake would make the bat too heavy to fly. So as the bat feeds, its kidneys are able to excrete large volumes of dilute urine, up to 24% of the body mass per hour. So having lost enough weight to take off, the bat can fly back to its roost and its cave or hollow tree where it spends the rest of the day. A combination of nervous and hormonal inputs regulates the osmoregulatory function of the kidney. These inputs contribute to homeostasis for blood pressure and volume throughout their effect on amount of osmoregulatory urine. Antidiuretic urine makes the antidiuretic hormone makes the collecting and duct epithelium temporarily permeable to water. An increase in the blood osmolarity above a set point, which triggers the release of antidiuretic hormone or vasopressin, which helps conserve the water. Decreased osmolarity can cause a drop in ADH secretion, which corresponds to a decrease in permeability of collecting ducts. Okay, so here the osmoreceptors in the hypothalamus trigger a release of antidiuretic hormone. This institutes thirst, causes thirst. Thirst causes the increased drinking of fluids. This will relate to homeostasis. The stimulus increases in blood osmolarity, which increases, releases the antidiuretic hormone. The osmoreceptor cells in the hypothalamus monitor the osmolarity of blood and regulate release of ADH from the posterior pituitary. ADH binds the receptor molecules on the epithelial cells in the collecting duct, leading to a temporary increase in the number of aquaporin proteins in the plasma membrane. Because aquaporin proteins form water channels, the net effect of the increased permeability of the epithelium to the water. To see how the response to ADH in the kidney contributes to osmoregulation, let's consider what occurs when blood osmolarity rises. As such, after we eat salty foods or losing water through sweating. When osmolarity increases above the set point, ADH released into the bloodstream is increased. The collecting duct's permeability to water rises, and the resulting is water reabsorption, which concentrates urine and reduces urine volume, which increases the blood osmolarity back towards the set point. Okay, this is the renin-angiostensin-aldosterone system. It also regulates kidney function. A drop in blood pressure near the glomerulus causes the juxtaglomerular apparatus to release the enzyme renin, and renin triggers the formation of the peptide angiotensin II. Okay, so a secondary regulatory mechanism acting on kidneys is the renin-angiotensin-aldosterone system, or a RAS. The RAAS involves the juxtaglomerular apparatus, a specialized tissue consisting of cells of and around the different arteriole, which supplies the blood with the glomerulus. Okay, basically, if you have low blood pressure, it's going to cause the renin to be released. That renin is going to be released. Is going to cause aldosterone, which is going to increase your blood pressure. I'm not very concerned about this one. Okay, Angiotensin II raises blood pressure and increases blood flow to the capillaries and the kidneys. Stimulates the release of the hormone aldosterone, which increases blood volume and blood pressure. Coordination of ADH and RAS activity. ADH and RAS both increase water absorption. ADH responds to changes in blood osmolarity. RAS responds to changes in blood volume and pressure. Thus, ADH and RAS are partners in homeostasis. Okay, here's this is on page 655. It's a description of small mammals, mice from Australia that are conserving water in their system. These are mice that were given water ad libitum, so whenever they wanted, they were given water. This is the concentration of urine and the molarity in their bloodstream. The concentration of urine was low. When they had no water available, these mice were able to conserve water by increasing the urine content in their blood. Basically, this is like if you ever pee, you'll notice that sometimes your pee is really dark and sometimes it's really light. That's just your body trying to conserve water. Okay, homeostasis. All right, stimulus and change, sensor, receptor, control center, response effector. Okay, these are how homeostatic systems work.